Hello, and welcome to Season 2 of the Canadian Epigenetics Environment and Health Research Consortium, or CEE-HRC podcast, presented by their trainees. The goal of this podcast is to chat about some of the explosive and infinitely interesting research happening in the realm of epigenetics, and particularly among the brilliant scientists here in Canada. We hope you enjoy this episode.
Hey everyone, it is my great pleasure to welcome our today's guest, Dr. Annie Ciernia. Dr. Annie is assistant professor in the Department of Biochemistry and Molecular Biology at University of British Columbia in Faculty of Medicine. Dr. Annie, I warmly welcome you to our today's podcast. Please give a brief introduction about yourself.
Yeah, thank you so much for having me. So yes, I started my lab at the University of British Columbia. I'm actually in the Center for Brain Health. And I started here in July of 2019. And so we had a number of months to get the lab up and going. And then we had a little bump in the road for COVID along the way. And now we're back in the lab working hard at all kinds of different projects that I'm excited to talk about today.
Yeah, how was it to start a lab during COVID? It must have been hard to find students, to recruit them, to train them when you couldn't be in the lab every day.
Yeah, it was challenging to say the least. And it was not what I expected when I started my lab. So when I was first starting, I, like everybody else, got lots of advice about how to start a lab and how to build a lab culture and how to make things work. And then that all went out the window almost immediately. And so I was fortunate in that I had a few months at UBC before the shutdown. So we were able to buy equipment. We had basic stuff in stock. I had a couple of students start, and then I hired a lab manager. And so we had a little bit of time together to build a bit of a foundation. Mm-hmm. But yeah, it was not easy. And then we spent almost 6 months outside of the lab doing some virtual things. I am a little bit fortunate in that I have a bit of a background in bioinformatics and computational biology. So we used that time, I'd like to think fairly wisely, and we spent a fair bit of effort building our bioinformatics pipelines. And we did some validation on some published data sets. I spent a lot of time teaching my lab folks a lot of the different tools that we would be using for their own data. So instead of the usual, like, do experiment, collect data, analyze data, we have been doing it in the reverse. We learned how to analyze the data, and then now we're collecting our own. But in some ways, I think it was quite helpful. It helps to give a better perspective on experimental design when you know what those output files should look like, what the QC should look like, what your statistical model should look like. So I don't think it was a waste of time, but it wasn't certainly what I had initially envisioned for starting my group.
Yeah, that's awesome. I think you used your time very wisely. So what made you interested in epigenetics or science, and why did you choose this specific field?
So maybe I'll tell you a little bit about where I grew up. So sure. I am from northern Minnesota in the States. I grew up in a very rural community. So the nearest town to my parents' farm is almost 10K away, and the town is 200 people, so very tiny. There were 19 students in my graduating high school class, to give you some perspective. So there wasn't a whole lot of resources or expectations for science, but I was extremely fortunate in that my dad was actually a research technician at the local university. And so he worked in plant sciences, and so he was in charge of running experiments in their greenhouse. And I got to go with him as a child to help water the plants, especially over Christmas break when all the grad students went home. My dad was very nice and went in to water everybody's experiments, and I was his helper. And I was just really struck by how amazing it was that you could have things like a control group and an experimental group, and then you could actually learn how something impacts an outcome. And for plants, it was very obvious at the time, like, oh, these ones were growing better, they're bigger, they're brighter. And I really fell in love with the idea of being able to do science. I didn't end up going into plant biology, but I really attribute that sort of early passion for science and discovery to spending many long hours with my dad in the greenhouse. I probably ruined like at least a dozen grad students' dissertation projects by helping, quote unquote, with watering or whatever I was supposed to be doing. But it really made a great impression on me.
Yeah, and how was it to switch from plants to brain? Like, was it a smooth way, or?
Yeah. So my undergraduate degree is dual degree, actually. So one in biotechnology, which was somewhat related to plant sciences, but was more just sort of like a general science degree. And the other one is actually in psychology. And so it
Oh,
happened actually a bit randomly. So I needed a job when I was a college student. So I was supporting myself. And I got a work learn position in a psychology lab. And it happened to be in a woman's lab who was studying cognitive neuroscience. And we were testing how eye movements can direct attention. And that was my very first exposure to research. And it just happened to be in a lab that was interested in brain science. And I really fell in love with that and trying to understand how the brain works. I was surrounded by all these cognitive neuroscientists. And there weren't many graduate students in that program. And so I had a lot of freedom and a ton of independence. And now that I have my own group, it's kind of terrifying to think how I was allowed to sort of have free reign in the lab, but it was an amazing experience. And I really attribute a lot of my love of neuroscience to some of those early mentors that I had. And I spent 4 years working in that lab, and I eventually decided that I was more interested in understanding how cells and molecules function in the brain that I was in trying to test human subjects. And so that's how I got interested in working more in molecular neuroscience. And so when I went to graduate school, I was looking for programs that sort of combined my psychology background and my biotechnology background into a more molecular focus of brain understanding.
did you ever felt like switching fields when you were younger, or?
I mean, I was pretty focused when I was young. I knew was really passionate about science, but I wasn't necessarily going on a straight research track. So I did consider other things. I actually took a scientific writing class when I was an undergraduate and really loved that. And I had thought at one point about going into like scientific journalism. I didn't end up doing that, but it is something that I do enjoy. And I think having a strong passion for writing also has helped my scientific career as well, especially now that I'm running my own group. I spend most of my time working on manuscripts and grants and other forms of writing.
That's great that you got a lot of diverse experience growing up and also during your undergrad that shaped your career. So what were the other major milestones that shaped you as a scientist during your PhD or postdoc?
Yeah, so I almost quit my PhD.
Oh,
To be honest. So I did my PhD at UC Irvine in the States. And I started in one lab and I was there almost a little over 2 years, and it didn't work out. Like the project wasn't going well. My interactions with my professor were not stellar. I wasn't progressing through the program. I wasn't prepared to take my comprehensive exam. Like it was not going well. And I almost walked away. And the reason I stayed was because the chair of the department took me aside and he said, "How is it going?" And I was like, "Oh, well, like I'm doing this and this and this." He's like, "No, really, how is it really going?" And I said, "Not very well. I'm not happy. I'm not progressing. I'm not learning what I should. And I'm not gaining the skills that I'm going to need to go on to do a successful postdoc." And he's like, "Okay, we need to find you a different lab to be in." And so because of him, I was able to switch labs to a different advisor, and the rest is history. It was really great. Like my second advisor and I really meshed well. We worked together well. I still am in contact with him regularly. And it really changed the entire trajectory of my scientific career. And it really made me realize how much impact a single mentor can have. Because if that relationship had not gone well, I think I would have quit and gone and done something else with my life. And so having that support and ability to really turn it around really made my career.
Yeah, it's very inspiring to have those kind of mentors in your life. And also it's good that your mentor asked you twice that how you're actually feeling, because many people just ask like one time, and because of shyness or many other things, you won't open up. But having that kind of attitude is very important. And it's great that you had that kind of mentor.
Yeah, and it's made me think a lot about how I mentor students in my own lab. And my interactions with them. I take mentoring very seriously. And making sure that the people in my lab are doing okay and that things are progressing as they should. They're making progress, they're learning things. The lab culture is strong and flourishing. And that's actually probably one of the most important parts of my job.
Yeah. Not everyone sees it like this. So it's really great that you would do this. I do think that also it's a crucial part of a PI's job to mentor and to make sure that everybody is happy and doing the right thing.
Thank you. It's not that they're happy all the time, but as long as they're productive and making progress but yeah, I do value my lab culture a lot.
Absolutely. It's great that you're making future scientists and future professors like yourself with great mentoring. So another comment was that your PhD was very successful. You had a Nature Neuroscience paper. So maybe tell us something about that.
Yeah. So when I was in Marcella Wood's lab, which was the mentor I ended up getting my PhD with, we studied how chromatin remodeling complexes are important for learning and memory formation. And I specifically worked on a chromatin remodeling complex called NBAF, which is the neuronal BAF complex. And I worked on a subunit of this complex that is only expressed in neurons. And it was originally discovered by Jerry Crabtree, and he did some work showing that it was important for neuron development. And we thought, well, since some of the same genes are potentially important for plasticity in the adult brain as in development, maybe this would be a potential regulator of gene expression for memory formation. And so in order to form long-term memories, what has to happen is you have to have new gene transcription. And what we showed was that this BAF53B, which is the neuronal subunit we were working on, we showed that this subunit is critically important for that transcriptional regulation to form a long-term memory. And so if you delete BAF53B or you make a dominant negative and you look at what happens to memory formation, you pretty much can't form long-term memories. What's interesting, however, is you can form short-term memories, which don't require transcription. So it's not that the animals couldn't do the task or couldn't learn something, they just couldn't consolidate it into a long-term memory.
Yeah, which type of assays are used to test the memories of animals? Because they cannot do like a writing exam or anything. What are the models?
So you can't just ask your mice what they remember about yesterday, but you can ask them sort of indirectly to show you what they remember. And so one of the main tasks we used was a relatively simple learning task in which you give the animals two identical objects to explore and they learn about them. And then the next day, you bring them back to the same environment with the same two objects, but now you move one of the objects to a new location. And so this is called object location memory. If an animal remembers the original training locations, they will specifically explore the object in the new location more than the object that's in the old location. So they have an innate preference for novelty, just like humans. So this was all done in mice. And so we can look for this preferential exploration of the moved object compared to the unmoved object. And then we can use those measures as a memory index of how well they actually remember the original location. And so animals that have disruptions of BAF53B really don't remember anything about the previous day in terms of where that object is or what the object is. We also looked at other kinds of memory. So we looked at some fear-related memories and other things, but most of the long-term memory tasks were impaired in these animals, indicating that you really need BAF53B and whatever genes it's targeting in order to form long-term memory.
Yeah, it makes a lot of sense.
So did you ever have a question in your mind that whether you want to choose industry versus academia after having a successful PhD?
Yeah, so after my PhD, I went and did a postdoc in Janine LaSalle's lab at UC Davis. And I worked on a bunch of different projects, but I was mainly focused on autism spectrum disorders and neurodevelopment. And I actually got interested in this because BAF53B and the NBAF complex are often mutated in neurodevelopmental disorders. And so I wanted to understand more about that side of the research. And when I finished my postdoc, I had a bunch of papers, looked really good on paper, but it was super competitive. I mean, that cycle, it was like 1 in 300 people in my area were getting jobs in academic science. So being realistic, I actually applied to a whole bunch of different areas of science. I applied to industry positions in R&D and also, of course, academics positions, to some private research institutes. I considered applying to some scientific writing jobs, although I was terribly unqualified for those, given what I had been working on as a postdoc, but I thought about it. And I interviewed at a number of different types of jobs. And ultimately, what I was really looking for was a solid work environment with good coworkers and something that was intellectually stimulating that I was excited about. And I think I could have found that at any of those types of jobs. It just happened to be that I found it here at UBC, but it wouldn't have had to be that. And I think I could have been happy at an industry job or something else, as long as the work was exciting and was challenging and well supported.
Cool, thanks. So before we go more into epigenetics, I have a question about life in general. So science could be stressful sometimes. So what is your major activity or hobby which took you out of that stress zone and help you in getting your highest productivity?
So that's a great question. And yes, science is often stressful, especially during a pandemic when things are not maybe as smooth as you might expect. So I have been a long distance runner for pretty much all of my life. And during the peak stress of grad school, I started hanging out with a bunch of my runner friends who were into ultra marathons, which if you don't know what that is, you should Google it. There are some really intense people out there. I never got that into it, but I have done a bunch of marathons, a couple of 50Ks, toned that back a bit now, maybe a marathon every couple of years or so, but I'm still out there running regularly. I have a group of friends that I'm out with every Saturday morning. It's sort of what keeps me sane, I think, and quite balanced. And it's one of the more important parts of my life. Even during grant deadlines and other types of things, that maintains. I don't sacrifice that for anything.
Yeah, I'm also a runner, so I understand you when you're saying that.
Oh, that's awesome. Yeah, I've heard from another professor also when I asked them a question about how they keep their sanity, and that's they have the same answer, running.
I highly recommend.
I need to start that. Thanks. So we will slowly transition to our next part, which is general public explanation of your research interest. What is your definition of epigenetics?
Yeah, so I think of epigenetics as the mechanisms that control how genes are expressed at its very heart. And of course, there's lots of nuances to this. So usually this is done in the context of a modification to the chromatin, which is the way you package the DNA into the cell, or a modification to the actual gene expressions, to the mRNA. And this is done also in the context of not changing the underlying genetic code. And so it's a way to use your genome in different contexts that allow the same genome within every cell to form different cell types, different tissues, different cellular responses to the environment. It allows us to be basically plastic to some level and adapt.
Yeah, thanks. And what are the aspects of epigenetics which you're using in your lab or research?
Yeah, so I have a project in my lab actually on BAF53B, looking at regulation of chromatin remodeling in the context of neurodevelopment, and specifically in autism spectrum disorders. We're looking at how BAF53B is impacting different types of neurons, and maybe it has different gene targets in different subsets of neurons. So that's one aspect. I also have a set of projects looking at microglia, which are the innate immune cells in the brain. And I got really interested in microglia when I was a postdoc, mostly because they kept popping up on all of these sequencing studies when people would look for differentially expressed genes in autism human brain samples. They would often find changes in neuronal genes, which was expected. And then there were all these immune genes that kept showing up. And at first, people just ignored them, thought they were some fluke of sequencing or who knows what. Yeah. But they became one of the most consistent findings across both the epigenetics and the gene expression in terms of impact or development.
Is there a lot of immune load in the brain? Are there a lot of immune cells, or is it varying a lot, or is it mostly stable?
Yeah, so it depends when you look. So microglia enter the brain, at least in mice, around embryonic day 9. In humans, it's like gestational week 4 to 5. And they actually come from the yolk sac and then they enter through the blood. So they're totally different than all of the other brain cells that come from a different lineage. And then they set up shop in the brain and they divide and they tile across the entire brain. And so they make up about 10% of brain cells, and they're responsible for basically surveilling the brain. They're looking for infection or damage or injury. And that's their main function. However, evolution has also co-opted them into a whole bunch of unique developmental functions. So they interact with developing neurons and astrocytes. They can help prune synaptic connections, so they can control how brain cells are connected to each other. They can help shape myelin, which is the sort of cells that help with synaptic transmission in the brain. They can actually engulf neuronal precursors and control how many neurons you get in a developing brain. So they have all of these unique functions in neurodevelopment that really have nothing to do with their immune function, so to speak. But because they're in this really unique brain niche, they have all of these additional interesting interactions with other cell types that we and others, of course, are super excited about trying to understand. Because if you alter microglia, as in the case of something like a neurodevelopmental disorder, they could have huge impacts on the rest of the developing brain.
Cool. That's great. So what kind of techniques you use in your research lab?
Yeah, so it's a real mix of things. So we have everything from neuronal and microglial cell cultures to animal models that we're working in. And so we go back and forth between the culture and the actual living organism. And we use everything from sort of basic molecular biology, biochemistry techniques to the sort of standard suite of sequencing approaches. So things like RNA sequencing, attack sequencing, chip sequencing. We're dabbling now in cut and tag and cut and run. And trying to integrate those, of course, bioinformatically. So we have a computational arm of the lab as well. And the ultimate goal is to really try to go from the molecules and genes all the way up to the level of behavior. So we have a full behavior suite that we can look at impacts on learning and memory and cognition, as well as all of the important neurodevelopmental milestones and phenotypes associated with neurodevelopmental disorders.
And when you do your RNA-seq or your attack-seq, do you do it in bulk, in single cell, or both? They both have, well, pros and cons.
So we've done both. So we've done some tissue things where you just really don't know what you're looking for and you're hoping to pull out a signal. We're moving now more to individual isolated populations. So we can isolate out neurons using flow cytometry. We can start to look at neuronal nuclei using similar types of approaches. We haven't done anything in single cell yet, but that is eventually, I think, where things will eventually head.
So based on the literature, how many types of clusters or how many types of cells are present in the microglia population?
That's a really great question. So of course, it depends how you do your clustering and what you're looking at. They're actually fairly heterogeneous early in development. There's a bunch of different populations that occur naturally. That becomes slightly more homogeneous as like you look at postnatal development. But there are differences between males and females in terms of sex differences in microglia gene expression. That's been shown by a number of groups. And also their responses in the context of inflammation in different types of neuroinflammation disorders. You get subsets that respond either protectively or detrimentally, and sometimes both at the same time. And so it's not completely clear exactly what drives some populations versus others. There's also differences in microglia depending on where you are in the brain. So there are some differences depending if you're in the front part of the brain that is important for decision making versus like in the hippocampus that's important for memory. So they're quite a diverse cell type that we're just beginning to really start to probe.
So if they're in a part of the brain that is more active than others, are their signatures going to be different?
That's a great question. So yes, there's some recent work just suggesting they can sense neuronal activity and even modulate it. So they can sense ATP release and they can potentially then in response dampen down sort of neuronal excitability. And so if you get rid of all the microglia, you actually become more seizure prone, depending on what you're doing.
Oh, so when they react to a pathogen or, well, when they carry out their immune function, can they affect the brain specifically because of that?
We think so. And I'm actually working on a grant to look at just that. So what happens in the context of neuroinflammation? What happens to the microglia? What happens to their interactions with neurons? And ultimately, does that contribute to some of these neuropsychiatric symptoms that we see in patients that have neural inflammation? So there's lots of open questions there, but we certainly think so.
Okay, it's interesting.
Awesome. So now we'll switch to next part where we'll have in-depth discussion about your research project or field. So what kind of models are you using for microglia in your lab?
Yeah, so we have a basic cell culture model in which we have an immortalized microglial cell line. We've also done some primary microglia cultures, and those are great ways to sort of develop tools, test initial hypotheses. But microglia, once you take them out of the brain, they are not like microglia in the brain. And so they're so finely tuned to their local brain environment that when you put them in culture, within a few hours, they actually start to change their gene regulation and their epigenome. And they actually start to look more like peripheral immune cells than they do brain immune cells. And so we can use these cell culture models to answer some questions, but ultimately we always try to go back into the brain in the native state to validate those. And so a lot of our experiments are in mouse models where we can make genetic manipulations to genes in microglia specifically at specific developmental time points. So we have transgenic tools to do that. And then we can take the microglia out, isolate them, and then look to see what happens to them specifically in response to different types of perturbations.
Is it known why they shift so quickly when you take them out of the brain? Is it because of the activity of the brain or a connection with neurons through receptors or signaling in the brain?
Yep,
Well,
a little bit of all of that. And so they respond to signals from neurons and growth factors from astrocytes. They're constantly contacting these cells physically in the brain and sampling those local environments. And so you disrupt all of that when you take them out. And people have gotten good at trying to mimic the brain environment. You can add in neuron released factors, astrocyte conditioned media, and they get a lot closer, especially now with some of the newer iPSC models. The medias have gotten quite complex and they can add in lots of additives and factors to try to mimic things, but it's often not 100% like they are in the brain.
Yeah, and have you tried to work with organoids, like the small brains that often we see in dishes that are spinning around?
Yeah, so the brain organoids are super cool. One of the things, though, is they don't have microglia. So all of the brain organoids are derived from a lineage of neuroectoderm that makes all the brain cells except microglia, because during development, microglia come from the yolk sac and they come in through the blood vessels later in development. So those progenitors are not there in most of the ways organoids are typically made. So what people have started to do is you can actually add them. So you take the yolk sac progenitors and you can add them right into your organoid cultures, and they will actually colonize the organoids to some extent, and they kind of look like what they do in the brain. So people are starting to do this, but the vast majority of brain organoids do not actually have them.
Yeah, so can that create a bias for those that study the organoids, since the microglia are not there?
I mean, there are lots of differences between an organoid and an actual brain.
Yeah,
You have the vasculature, you have the connection to the rest of the body. So microglia are just one of them. But yeah, I mean, they're almost 10% of the cell types in the brain. And so you're missing them from most of those contexts. So, and when people have started to add microglia in and looked for impacts on the rest of the organoid, it does have impacts, which is not surprising. So you can get an answer without them, but it might not be the same answer that you would get with them.
So how much of that impact is epigenetic on the neurons if you co-culture them with microglia versus without?
Yeah, that's a great question. Not well known. And so we know that microglia can signal to neurons and vice versa. And that neurons in distress, of course, can signal to microglia, and that causes them to become reactive and try to fix things. But it's not as clear what epigenetic impacts you might have on neurons or astrocytes if you have abnormal microglia. Like those types of interactions aren't terribly well described, and the long-term impacts on cellular function are not as well known. And it's a little hard to tease them apart from direct impacts, right? So say you have something like neuroinflammation. Well, that has impacts on all of the brain cell types, and then they're all talking to each other. So it's a little tricky to figure out what's driving what. But yeah, it would be super interesting to try to pull that apart.
Sure, thanks. So is BAF53B also expressed in microglia?
It is not, no. So we see the non-neuronal homolog there. So there's BAF53A. Yeah, and we haven't looked at astrocytes. We should do that. Supposedly, in all of the single cell stuff that we've looked at, you don't see BAF53B outside of neurons. And it does appear to be in all of the different neuronal subtypes. So
Cool.
it's fairly unique.
Yeah, is there any microglia specific regulator or BAF, which is in microglia only?
I would love to know the answer to that. If anyone does, please contact me and I would like to work with you. Yeah, I've thought about doing that experiment. Yeah, I mean, of characterizing by like mass spec or something like that. Nothing is known in terms of a specific remodeler for microglia.
Cool. So one more question about the models, comparing mouse model to human model. There's some species specific differences. For example, in enhancers also, there was one paper on H condyles. So there were some non-coding elements which are deleted in humans, but conserved in mouse and chimps and dogs even. And one of the gene was GADD405G and was concluded that maybe it helps in the brain specific expansion. So is there a way we can make these mouse models or cell models more closer to human models? And how far we should go for these model developments to make it as closer to human, or is it even needed? Maybe not for fear conditioning, but what are the other questions which are not answered by the mouse models?
Yeah, I think it depends what kind of question you're asking. So if you're asking about gene regulation and cell biology, I think the organoids might be a great way to go, right? Because you could make them from human cells and you would have all of these human specific regulatory elements and look at how they control gene expression. If you're interested in what those regions do in terms of behavior development or cellular development in an endogenous system, that's much trickier. I mean, people have started to introduce like a single enhancer element from humans into mouse and look to see what happens to other types of phenotypes. So you could imagine testing cognition in that context and see if it changes. But that's like taking a brick and throwing it at a house and trying to understand how the house works. Like that's pretty difficult. So I think there will be limits to what rodent models can tell us about some of these primate specific regulatory elements. And I mean, people have started, not as much here, but have started looking at macaque models and other types of primate models. There are a couple of different models now for Rett syndrome that have been done in monkeys. I think they're in Japan, but trying to understand some of these more primate specific sorts of regulation in the context of neurodevelopment. And some of these things just might not be answerable in a model outside of that.
That's great. Has there been any epigenetic therapy which uses the microglia or neuronal cells?
So not for microglia, but there are people interested in trying to either silence or reactivate different types of genes that have come up in neurodevelopmental disorders. So there's a bunch of work ongoing right now for Fragile X syndrome to try to change the expression of FMRP. Also for Rett syndrome, there's been a lot of interest in gene therapy and trying to fix the MECP2 locus that is mutated in that disorder, and also Angelman syndrome. So that's an imprinting disorder in which you can try to reactivate the other allele. So yeah, there's lots of interest in this. Most of it is moving forward in animal and maybe even some primate models. So we're maybe not too far off from eventual clinical testing. This is certainly a hot area, but it's tricky, right? So eventually you want to do this in children and you don't want to make them worse. And so you have to be fairly confident that what you're doing is not going to be disruptive, even more so to neurodevelopment. The brain is also very difficult to target therapeutically.
Yeah,
it's hard
because of
to access.
Barrier.
Yes. I mean, in animals, we can do it with AAV, so adeno-associated virus. You can infect neurons and they're very safe, and they have pretty high efficacy. And there are some approved AAVs for like treating eye disorders. So which is part of CNS. So there is some movement along that, but I mean, you would have to be able to deliver it into folks, probably in through the spinal cord and get it into the brain. The other tricky thing is knowing when you would give this. So most clinical trials don't start off in children, right? You start off in adults or adolescents where you're going to potentially do much less harm if something goes wrong. But that might be a really difficult point in time at which to correct a neurodevelopmental disorder, because you've had a lifetime of abnormal brain development, and you can't just wipe the slate clean and start over immediately. So it might not work in older people as well. So it'll be a difficult road to go down, but I think it's so worth doing. Like if you could offer something for parents and families that have children, especially with severe neurodevelopmental problems, to be able to offer them something, I think is a really good goal for all of us to have.
Yeah, it would be an amazing way to cure so many diseases that affect children and adults too, because they have it their whole life.
Yes, these things don't go away.
Yeah. And how are the models different in terms of timing for the development of the brain? Because if we want to go from a model like a chimp to humans, we probably need to administer the drug at a specific time. So how similar are the times, or just a different scale, or the timing is completely different?
Yeah, it's tricky. So there's lots of different milestones you can look at in development in terms of brain size, brain region formation, cell integration, when, for example, microglia are pruning synapses. And people have done a lot of work trying to compare mouse to rat to primate to human. And so there's a fair bit of work on this, but it's never a one-to-one, right? Like a 3-day-old rat is not exactly the same as a 3-year-old human. Like there are going to be differences. And so, yeah, that question is a bit tricky. And you also have to think about what's actually feasible in the clinic, right? So most kids with autism aren't diagnosed till they're 3 to 5.
Oh, yeah.
So for microglia, how close is their epigenome to the other immune cells or macrophages in humans or in mice?
Yeah, no, that's a great question. And that's been profiled both in humans and in mice. There's human microglia data from postmortem brain that's out there. And they're somewhat similar, but distinct enough. And so there was a fair bit of work in the mid 2000s looking at how different types of macrophages adapt to their local tissue environments. And so this isn't just unique to microglia. You have tissue resident macrophages in pretty much every tissue that encounters the environment. So in your lungs, in your skin, and each one of these different macrophage populations adapts its gene expression and function and its epigenome to the local environment, including the microglia in the brain. So they have a bunch of unique enhancers that turn on a bunch of unique genes that only microglia express in the brain. And people have done some really clever experiments where you can transplant tissues or transplant the macrophages from one tissue to another. And so if you take like skin macrophages and you put them in the peritoneum, they can kind of adapt, but not completely. So there's some programming that happens really early and then some adaptation that occurs in signaling to the local environment. So it's a mix of both.
So can you epigenetically reprogram these macrophages and make it more microglia-ish and then implant in the brain and see if it works similarly or not? Has it been done?
You can. Yep, that has been done. So you can actually irradiate the brain and all the microglia will die. It's kind of hard on your brain, but I mean, in rodents, they survive and you get rid of all the microglia. You can also deplete microglia with a drug that it was made by a company called Plexicon, but it's an inhibitor that blocks a survival receptor in microglia. And so most of them will die off.
Cool.
And then you can actually transplant in macrophages from the periphery. And they will populate the brain. They will tile across the brain. They look surprisingly similar to microglia. Transcriptionally, they're similar, but not identical. And so they can adapt, but they're not exactly the same. And there are some differences in function that are, I mean, people haven't probed every possible thing you could do after this to see what happens, but they do respond somewhat differently than if you just had the endogenous microglia. There are also some cool experiments where you can take microglia from a female mouse and put them in the brain of a male mouse. and you can look to see what happens. And so there appears to be some sort of long-term programming of microglia depending on sex. And when you transplant them into the other brain, so when you swap them into the other environment, they don't lose that. So they adapt somewhat, but you can still see that they are adapted from the other sex, which is super cool. Like, so there's some reprogramming that's happening, whether it's hormonal or sex chromosome driven. It's not completely clear, but super interesting.
Yeah, and it has to happen really early so that it doesn't change even if you change it. Well,
yeah, it's thought to happen when you have some hormonal surges during early development.
Oh. Are there any ways to screen for a disease of the brain before the child is born to?
Yeah, it's a great question. And one that, of course, there's a lot of interest in. The tricky thing is that all of the genes that have been identified that have been linked to autism, there's a lot of them, like hundreds, maybe even thousands if you go to some of the really rare ones. But even the most prevalent ones are like 1% of autism kids will have a mutation in gene X. So they're rare to begin with. And right now, they're not really actionable. And so there's not, if you screen, there's nothing you could do to fix that right now with the way therapies are set up. So they're not screened for in most cases. If you were doing something like whole exome sequencing on embryos or something, you could screen and pick those up, but that's not common clinical practice as of right now.
And how clear is it that the mutation is going to lead to the disease or to some point on the spectrum?
Yeah, that's also the big trick, right? So there are some known mutations, like for example, in Rett syndrome, if you have one of the common mutations in MECP2, you will probably have Rett, but not always. So especially in the case of something like Rett, it's on the X chromosome and you have random inactivation. So you may have very severe disease or less severe disease, depending on random inactivation. So a lot of these things are not as deterministic. And we also don't understand what a lot of these genes do. And so some of the variants may do nothing. We don't know. And there are also cases from some of the larger human consortium genome studies that people are like homozygous knockout for something that looks like it should be really detrimental and really bad, and they're totally fine. And they were in the control groups just getting sequenced. So we really have a long way to go, I think, before we could make deterministic sorts of judgments on at least a lot of the variation that we have. And I mean, the autism genetics is complicated by the fact that, say you have a child with autism and you do exome sequencing, only about 30% of the time do you find a variant that clinicians are fairly confident caused the problem. Most of the time, they don't find anything. So there's a lot that we still don't understand about how this works.
If the screening was more about epigenetic changes, would it be better? Do you think?
It depends. So there are rare examples, like something like FMRP, where you can get silencing that is epigenetic. But most of the epigenetic signals are even more complicated than the genetic ones and potentially even less deterministic. However, I mean, people are certainly interested in biomarkers for neurodevelopmental disorders. And if some of those genes were epigenetically modified in such a way that was actually causative, it might be easier to correct, right? So that might be more malleable than something like an actual genetic change, but that's far less well understood.
Yeah, I had the same question, that what is the non-genetic component, either epigenetic?
Right. So I did a lot of work in my postdoc looking at environmental impacts and risk factors for autism. And there's a lot out there. So infections during pregnancy can increase risk for autism and neurodevelopmental disorders, but it's only a small percentage of increased risk. So it's not clear exactly how that works. But there are lots of environmental factors that are thought to potentially contribute, probably on top of some sort of genetic risk factor.
Could be a mix of, if you have these genes in this type of environment.
Exactly.
Yeah, and better decoding of SNPs need to be done, which is in non-coding genome, which is hard.
Yep, and that's actually where the autism genome sequencing is going. They're now looking in the non-coding genome. It just takes longer and you need more power because there's more variability.
So what are the major challenges in your field moving forward from here?
So, I mean, one of the biggest issues in looking at epigenetics in the brain is just the sheer diversity of cell types that are present. And I mean, that has come a long way with a lot of the single cell approaches, but trying to understand and link it back to the underlying biology is not trivial, especially when you're dealing with rare subsets of populations. How do you identify them? How do you manipulate them? How do you manipulate gene in the brain? And it's a lot harder to do causational experiments, right? So if I'm trying to identify a classic gene target, then I want to go back and manipulate it in the brain. Like, what if there's not a transgenic mouse available? Do I have to make a transgenic mouse? I then have to do the conditional deletion in my cell type of interest. And 2 years later, I can maybe answer my question of, is this gene important for something in this cell type? So it can be a lot slower. And that's where the cell culture models, even if they're not perfect, can be really valuable, because you could have an answer much quicker, but it might not be the same answer that you get if you actually did it in the brain.
Are there any humanized mice with the patient-derived brain cells or neuron cells?
So people have done a lot of these brain organoids from human cells, and they have put them into like the peritoneum of mice to look at vascularization. I'm not aware of any Frankenstein mice where you can actually switch out the brain. And even some of the more mature organoids, I mean, they start to have electrical and synaptic activity that almost becomes a little sci-fi at some level. So I don't think anyone has done that yet.
Okay, awesome. Thanks. So before we let you go, do you have any words of wisdom for the listeners?
I guess one of the things I would suggest, especially to trainees and students, is it can often be difficult. Grad school is hard. Postdoc was hard. There are some real low points. But there are also some really amazing high points. There is nothing quite like being the first person to see a new data set come out and make a new discovery. And you just have to hold on to those high points to help see you through the lows. And everybody goes through those low points. You're not alone. And don't be afraid to reach out to your coworkers or your cohort and talk about these things. I realized when I was a graduate student that there was not really this culture of talking about failed experiments as much as there should have been, because we've all had them. Everyone does. Most experiments fail. Yeah. It takes 10 times to get something to work. And that is standard. That's the way it is. It's not that you're not good at it. It's just that is the nature of the game. And so especially like things have been pretty difficult for a lot of folks over the last couple of years. But hang in there. I'm confident we're going to come out of this and science will go on and it'll all work out.
Yeah, well, these are some really great insight, all the knowledge. It has been great.
Thank you so much. Podcast was awesome.
Thank you guys for doing this. It was a lot of fun.
Thank you for listening to season 2 of the This is Epigenetics podcast. If you like this podcast episode, you can check out our website and future episodes at thisisepigenetics.ca. We hope that you all have a very good day.